Okay, so with this video, what I want to do is go over some examples of enzymes and really hit onto this idea of anabolism or catabolism. So with anabolism, we are going to synthesize or build larger molecules, and with catabolism, we're going to go through decomposition or breakdown of larger molecules. So anabolism is building up, catabolism is breaking down. You may not have heard those two words before, but you've probably heard of metabolism before. And when we talk about metabolism, it's the sum total of all the chemical reactions that are taking place in someone's body. And if your body is doing more chemical reactions, if you're breaking down more things, if you're um, generally processing more um, molecules for energy, responding to your environment more, you're going to have a faster metabolism. Okay, so different environmental conditions can affect that as well. If you're outside on a cold day, you're going to have a higher metabolism just trying to stay warm because you're going to be breaking down more molecules for it to generate heat, and your muscles might shiver, and you're going to ultimately just be doing a lot more chemical reactions. So as we go through this, it's going to be a lot of information. It's going to be a lot of words. See if you can get some of this out of here. All of these details are obviously not necessary to understand at this point, but see if you can learn something. Okay, so we're going to start out with catabolism, or the breakdown of larger molecules. And most of the time when people hear of enzymes, they think about digestion. And digestion begins actually in the mouth, chemical digestion, where your saliva actually contains enzymes that begin the breakdown of carbohydrates. But from your mouth, your food heads down your esophagus into your stomach. And from your stomach, it heads into your small intestine. And the first segment of your small intestine is the duodenum. We have other things that are producing, like the tube itself contains enzymes, but we also have enzymes, especially from the pancreas, being shot into the digestive tract to break down your food that you eat, the molecules in your food, small enough so then they can be absorbed when we get into the jejunum and the ileum in the small intestine. Ileum enters into the cecum, into the ascending colon, the transverse colon, which the colon we're thinking of as our large intestine. We're removing water. There's bacteria in your large intestine that are processing fiber and making vitamins for you. You go down your descending colon to your sigmoid colon, to the rectum, and out the anus. So if we are imagining this inside the small intestine, there's digestion and absorption taking place. Our next unit, we're going to be talking about transport across membranes and how things get through cells, into cells and out of cells. And but if you ever see something like this, when we look at all of these different models, scientific models, if you see something going in with an arrow and the arrow pointing out to something different, a chemical reaction has probably occurred there. And if it's inside a living thing and a chemical reaction takes place, there's generally an enzyme that's going to be associated with it. So maltose is a sugar, a disaccharide, because it's made up of two monosaccharides, but oses are sugars, and aces, if it ends in ace, it's probably going to be an enzyme. So as we progress here, I don't want you guys to forget about this lock and key model, because specific enzymes are designed for specific substrates. It's not just we have one type of enzyme that does all the chemical reactions in our body, and they have to be the right shape. So protein digestion begins in your stomach. So we begin a little bit of carbohydrate, and a lot of people think of stomach acid breaking down your food. The reality is that it's the enzymes that are breaking it down, not the acid. The acid does, this hydrochloric acid with all these extra hydrogen ions floating around inside your stomach. The cells lining the stomach will secrete the enzyme into the stomach, and it's the hydrogen ions that will make it the right shape so then it can fit with the substrate, which would be eaten proteins, and break them down into polypeptides. And then this would progress towards other enzymes that would be the right shape for the polypeptides to break them into peptides and then ultimately into amino acids, which then are small enough to be absorbed into our bloodstream. Now, again, we think of this lock and key model, they have to be the right shape. We want to break down the proteins that we eat, but we don't want to break down important proteins within these cells, and therefore it's kind of started as this enzyme is made in a shape that is inactive. Okay, when it comes into the right environment with the right pH, then it can become active and it can do the substrate can bind to the active site. So again, this general model of we have an enzyme, we have a substrate, the substrate lands on the active site, chemical reaction occurs, and products are formed. This is showing a larger molecule becoming two smaller molecules, and the substrate is sucrose, the enzyme is sucrase. This would be a decomposition reaction, and so this would be catabolism. As we go into digestive or catabolic enzymes, we have lots of these, okay? And they're coming from different locations in your body. And they're breaking things down smaller and smaller and smaller and smaller and smaller and smaller until they ultimately can get through the channels or pumps in the cell walls of our small intestine and into our bloodstream. So if we start at the mouth, we've got our salivary glands that are shooting amylase into the mouth, which will break down our amylose into disaccharides or smaller sugars. We go past the pharynx, down the esophagus to the stomach. In the stomach, there's pepsin, which is an enzyme, and it doesn't end in ase, but it's still an enzyme, and it's going to initially break down the proteins into smaller polypeptides. The liver here, and we've played with liver with our hydrogen peroxide lab, the liver is going to make bile, which is stored in the gallbladder, and it's not an enzyme, but what it does is it separates the individual fats, so then the fats can actually be accessed by the enzyme because we need the fats isolated. They can't be one big glob of fat. And the pancreas here, and this is, I want you guys to kind of understand here how important your pancreas is. Sometimes people think about your pancreas and they know it has to do with insulin and diabetes and all of that kind of stuff. But the pancreas has kind of a dual function. Not only is it an endocrine gland that secretes hormones like insulin, but it's really important for producing enzymes for digestion. And because it's such an important organ, um, a lot of times it's difficult to treat pancreatic cancer, and if you can't really live without your pancreas. So pancreatic cancer is one of the more deadly cancers. So your pancreas is making lipase, which is going to break down lipids or fats into fatty acids and glycerol. It's also producing more amylase because the amylase that started the digestion here, once it hits the hydrochloric acid, that amylase no longer works. So your pancreas needs to reshoot some amylase in there to continue to break down the carbohydrates that make it through your stomach. The pancreas is also making trypsin, which is the next enzyme that's in the protein section that's breaking down the polypeptides into peptides. You hear peptide, that associates, or amino acids, that all associates with proteins. So now we're in our duodenum here, which is the first part of our small intestine. The duodenum itself secretes maltase, sucrase, and lactase, which are breaking down the disaccharides into monosaccharides. The duodenum is also secreting enzymes that are peptidases, which break down the peptides into amino acids. As we keep going, it's more about absorbing nutrients, absorbing water, and ultimately getting everything that we need to keep out. Okay, So if we're looking at this giant chart, we've got amylase, we've got pepsin, we've got lipase, we've got trypsin, we've got maltase, we've got sucrase, we've got lactase, we've got peptidase. So all of these ACEs and trypsin and pepsin, they're all catabolic. They're all breaking things down. You can see in this chart the substrates and the products that are being produced and where they are taking place. Now, if you're using this video to try and get some notes so that you can be advanced on your assessment, this might be a time to pause it and write a couple things down. I know you guys have had this chart that I asked you to fill out before, but this is basically your answer key to that. All right, so I'm going to keep moving on. That was all the catabolism stuff. So I want to talk a little bit about anabolism, or building up. There's an enzyme on the chart that's called glycogen synthase, and synthesis reactions are building up, and the enzyme name kind of tells you what is being built up. So glycogen synthase, it's an anabolic reaction that takes the excess glucose from what you ate and builds it up into glycogen, which is a stored energy source in your liver and muscles. So once you begin your digestion, right, we're absorbing all of these nutrients, and all of the blood that is absorbed and packed full of nutrients initially goes to your liver before it heads up to your heart and to your lungs and to the rest of your body to send those nutrients to every single cell in your body. Every single cell needs nutrients. But your liver is going to do some processing. If there's dangerous products that have been absorbed, your liver is going to try and neutralize those and break them down. It's also going to store up for later. So if you guys have ever heard of carb loading or carbo loading, you know, people eating a lot of pasta before a big race. It's basically topping off your liver and your muscles with glycogen, because when you haven't eaten in a while and your blood sugar goes down, you'll have hormones that recognize that your blood sugar is low, that will ultimately talk to your liver and bring this stored glucose back out. But the process of storing it, and remember when we're playing with those livers, it kind of looks like lobes and they have these little holes in it. Those holes are from these central veins that are inside um, all of these cells that are packed together here in your liver. So while we were just playing with chicken and calf liver, it's the same basic anatomy with a human liver. And it's in these cells where, as this blood flow is high nutrient blood is passing across these cells, the glucose will transport into the cell. You see the cell membrane. It'll go into the cell, and then we'll have ACEs. You see hexokinase 2, which takes the initial glucose and builds it up a little bit bigger, and then glycogen synthase links them all together. Okay, so then we end up with this glycogen molecule, which is a complex carbohydrate that we can store and break down later. So this is a anabolic. It's building up. Glycogen synthase is taking glucose and building it up into glycogen, and that's going to take place mostly in your liver and your muscle cells. So we have all this glucose, and we use our glucose as energy. Our carbs are energy, right? So this is going to get us into cellular respiration. So in this, we're going to talk about some things that we'll hit again later in the year. But we can't use what we talked about before, is that this energy, it's like gold. Glucose is like gold, but we can't spend gold at stores to pay for the stuff we want to buy, right? We have to have cash. So we have to go through this process in our mitochondria in our cells where we take the gold, we take the glucose, and we break it down, and we transfer the energy in the bonds of the glucose, and we transfer that energy into the bonds of ATP. So we're going through this catabolism, or breaking down of glucose, and we're doing this anabolism, or building up of ATP. And then when we need to do work inside of our body, whether it's muscle contraction or pumping things across membranes to make your brain work, we then ultimately break down ATP to release the energy from ATP. So this process of breaking down glucose, on the overall chart, I did not list the enzymes that are associated with glycolysis. Now, when I teach you about glycolysis, we have 3 steps of cellular respiration: glycolysis, the Krebs cycle, and electron transport chain. When we learn glycolysis, we just talk about taking the sugar, splitting the sugar, getting a little ATP, carrying some electrons, and moving these split sugars inside the mitochondria for the Krebs cycle. That's all we really need to know about glycolysis. However, if we're really going to get into glycolysis, it's not just one splitting of the sugar. There's 10 steps to glycolysis, and every transfer of the um, carbon structures here, ultimately we're going to get 2 pyruvates, which are 2 3-carbon molecules. But for every step, all these 10 chemical reactions that it takes, it's not just one splitting. There's a separate type of enzyme that's going to do every single step here. So while my list for enzymes seems like a lot, there's so many more enzymes in reality in living things, because every single chemical reaction typically is going to be associated with an enzyme to speed up that chemical reaction. Same thing with the Krebs cycle. When I am going to teach you about the Krebs cycle in here, we take the split sugar, we break it down into carbon dioxide, which leaves the mitochondria and ultimately will leave the cell to the bloodstream and out your lungs. We're going to make a little ATP and we're going to carry lots of these high energy electrons. That's what I'm going to tell you for the Krebs cycle. But if you look at this Krebs cycle, it can be almost infinitely complex. And every time you see an ACE, it's another enzyme doing another chemical reaction. So there is a tremendous amount of things going on here with the chemical reactions in biology, and I'm just going over a couple examples here. Now, we said with cellular respiration, the idea is that we break down sugar, we take the energy from the bonds of the sugar, and we transfer that energy into the bonds of ATP. So as we look at this, at the very end in the electron transport chain, we have this enzyme, and it's one of the enzymes I really need you guys to know because it's really important for photosynthesis and cellular respiration, because this is where we generate almost all of our ATP. And again, you see a molecule or molecules coming in, an arrow coming out, and something different coming out. So this is going to be an ATP synthase molecule. These protons or hydrogen ions will go through it, and that triggers this enzyme to build up or synthesize ATP. So ATP synthase enzyme is anabolic. It's going to take a DP plus an extra P and make ATP. The D is di for 2 phosphates. The T is tri for 3 phosphates. And again, we're going to have this in the mitochondria in every cell in your body, but in any kind of eukaryote, anything that has a nucleus, is going to have ATP synthase. ATP synthase is also in all the mitochondria in plants, but also in the chloroplasts of plants. ATP synthase is in the outer cell membrane of bacteria. So every living thing uses ATP as energy. In order to generate ATP, it needs ATP synthase enzyme. It's one of the most abundant enzymes there is. So it's pretty darn important. Now, get a little bit crazy here, because this is ultimately why this is a little bit of the science behind our catalase that's in our liver that we were playing with in the hydrogen peroxide. So we were just saying here that ATP synthase is making ATP. Now, in order for these hydrogen ions to come rushing back, they need to be pumped across this. So this is a pump, okay? So these electrons are using their energy to push these protons across this membrane inside the mitochondria. I know I'm speaking a whole nother language here, but just try to stay with it as best you can. All of this stuff is going to come back around, and the more times you hear it, the more it'll start making sense, even if it doesn't at this moment. So, oxygen is part of cellular respiration. It's going to clean up electrons that have been used to pump the hydrogen ions across. As it picks up electrons, it's going to grab some protons, which ultimately is picking up hydrogen. Now, in order for oxygen to become safe for your body when it picks up electrons, it needs to pick up 4 electrons in order to get to water. If it does not pick up and clean up enough electrons, it does not become water, it becomes one of these other things like hydrogen peroxide. Now, these are called free radicals or reactive oxygen species, and they can be very dangerous. So these things, they still want another electron even though they didn't get it initially. So it's they're not just going to take the electron and wait around at the inside the mitochondria. They're going to go out and they're going to go and steal electrons from wherever they can find them, which may be the lipid bilayer. And once it steals an electron from our tails, those tails are stealing from its next tails and next tails and next tails, and ultimately you're destroying cell membranes. We can also steal from proteins and DNA. So when we have these reactive oxygen species or too much hydrogen peroxide in our system, we can ultimately cause mutations because they can steal from the DNA, and that can ultimately cause cancer. This oxidative stress from these too much hydrogen peroxide in your system ultimately is linked to a tremendous amount of diseases the more and more research that's taking place. So, how do we prevent getting sick from having too much hydrogen peroxide in our system? Because we're constantly making it as we're trying to generate ATP. And we have catalase and peroxidase. And catalase is what we were talking about with our liver lab. That's the enzyme in the liver that's breaking down the hydrogen peroxide into safe water and safe oxygen gas. So inside of our cells, we have these mitochondria that at times are creating these reactive oxygen species like hydrogen peroxide. And then we have the enzymes inside peroxisomes that grab that hydrogen peroxide and turn it into water and oxygen and make it safe. So another type besides catalase is called peroxidase. Again, the enzymes are ending in ase. They're catabolic. They're breaking down hydrogen peroxide into water and oxygen. Again, these peroxisomes are in every single cell in your body because every single cell in your body needs mitochondria to make ATP because every single cell needs energy. But we have a lot higher concentrations of peroxisomes in our liver, so that's why we used liver, but we could have used any source of cells in order to do it. And some of you guys with the lab, you used yeast and you used different stuff in order to be a source of enzyme. But catalase and peroxidase break down hydrogen peroxide into water and oxygen. Since they're breaking it down, it's catabolic and it's found in the peroxisomes inside your cells. Okay, now we've talked a lot about breaking things down, a lot of catabolism. So I want to switch over to anabolism for a little bit. Now, if we have one cell, sometimes cells get damaged. All of us, even though we're trillions of cells, began as one fertilized eggs, as one cell. And we had to go through this process of the cell cycle to grow and repair cells throughout our life. So we go from one cell to two cells. And there'll be another unit on the cell cycle where we talk about how we get from one cell to two cells and how we divide all these chromosomes, because the DNA in one cell we need to make a copy of all that DNA to make two identical daughter cells. So in order to make two cells, we need to make a copy of our DNA, and that happens in DNA replication. And again, DNA replication is going to be a chemical reaction. So when we make a copy of all of our DNA, we use an enzyme to help with that. And DNA polymerase, it's DNA, it's a polymer, it's helping make a big molecule here. So it's an enzyme that's helping do that. This DNA polymerase, it's anabolic because it's building up. It's taking these individual free nucleotides and hooking them together in a sequence here to build up a giant separate DNA molecule. So ultimately you'll have a DNA molecule here, you'll have a DNA molecule here, and each of the new molecules is going to be half of the original strand. So DNA polymerase is an anabolic enzyme that takes free nucleotides and builds them up into DNA in the nucleus of your cells. We also have transcription. So when we're learning our functions of our macromolecules, when we said nucleic acids, they store genes, which is in the case of DNA, and they make proteins, which is in the case of RNA. In order to make proteins, we first need to generate a messenger RNA strand, and that happens in the nucleus. And instead of DNA polymerase, we're not making more DNA, we're making RNA. So RNA polymerase is this big enzyme here that's helping unwind the DNA, and then it's grabbing onto free nucleotides and it's attaching them together to make a copy of a single recipe of a gene. So it's not copying the entire DNA strand, it's only copying a section that is going to end up being the recipe to make a specific protein. So RNA polymerase, it's anabolic, it builds up RNA from free nucleotides in the nucleus of a cell. Okay, I just want to talk about plants because we talked about humans and people and everything, which a lot of the things I've been talking about can apply to other organisms. But let's talk about plants for a little bit, because if we didn't have plants, we would not be alive, okay? Life as we know it would not exist without plants. So in this little model here, it says photosynthesis. The plant draws up water from its roots. It takes in carbon dioxide from the air. The leaves trap energy from the sunlight. So all good so far. Step 4 says the plant uses the energy of sunlight to turn water and carbon dioxide into sugars and oxygen. That's true, but that statement is at a level that's a little bit below where we're going to get, because it's not like the sunlight turns water and carbon dioxide into sugar and oxygen. There's chemical reactions that have to take place to take water and carbon dioxide and take the building blocks of those molecules and convert it into glucose and oxygen. And if those chemical reactions are taking place, it's ultimately enzymes that are essential to those chemical reactions and photosynthesis. So when we start looking at photosynthesis, we break it down into two different things. When we do cellular respiration, we have 3 steps. We have glycolysis, Krebs cycle, and the electron transport chain. When we talk photosynthesis, we break it down into the light-dependent reactions and the Calvin cycle. So water is going to come in for the light-dependent reactions, and it's going to break down, and oxygen is going to be produced. This is happening in the chloroplasts of the leaves of cells. Photosynthesis doesn't take place in every single cell in a plant. It's not taking place in the roots or in the bark. It's only in the green parts like the leaves. So the Calvin cycle brings in the carbon dioxide and we get glucose out. So there's 2 big things here. You see arrows where you have a substrate coming in and a product coming out. You have arrows where you have substrate coming in and you have product coming out. So there's got to be enzymes involved in this because it's a living thing. So if we're looking at these light-dependent reactions, again, we're going to get into this. There's this really complicated name for an enzyme here, water plastoquinone oxidoreductase. Okay, it's going to be catabolic. It's going to break down water. So where a lot of people have this a little bit wrong is they say light breaks down the water. Light is not breaking down the water. Light is energizing these electrons, and the electrons are powering these pumps that are pushing these hydrogen ions across the membrane. It's an enzyme here that's ultimately breaking down the water to produce extra protons or hydrogen ions and give the electrons to this chlorophyll. This molecule right here is a pigment that has the ability to capture light, and this is what makes plants green, these chlorophylls. So this enzyme breaks down water to make oxygen and hydrogen ions and electrons here, and that's in the chloroplasts within plants. Now, we also, remember back to ATP synthase, right? We had ATP synthase in cellular respiration, but here also we have ADP becoming ATP. So ATP synthase is the enzyme that generates ATP. This is also happening in chloroplasts, not just mitochondria. ATP synthase is anabolic because it's building up ATP. Now, this last enzyme I'm going to talk about here is the most, they say it's the most abundant enzyme in the entire world. Okay, so it's called Rubisco. It has a really long name, but we just call it Rubisco. And again, it's so incredibly important because it takes carbon dioxide from the air and captures it, and we are able to make sugar because this enzyme is able to take the carbon dioxide from the air and fix it into a larger molecule. So this Rubisco enzyme is anabolic. It's taking carbon dioxide as a substrate and it's building up a 6-carbon molecule. We're going to say glucose. Calvin cycle again is one of these really complicated, like Krebs cycle thing where there's lots of intermediate molecules, but we're just going to say it takes carbon dioxide and makes glucose. This is going to be in the chloroplast. Again, basic idea here, carbon dioxide comes in so we can make sugars. So the full name here is ribulose 1,5-biphosphate carboxylase oxygenase. So that's a mouthful. So I think the longest word in the human language is the name of an enzyme. So a lot of times these get abbreviated. So Rubisco here takes the carbon dioxide and builds it up into a 6-carbon molecule. So it's right here. It's a catalyst. It fixes carbon. So carbon fixation is when we take atmospheric carbon dioxide and turn it into ultimately an organic macromolecule, which these G3P ends up making glucose. But not only does this G3P end up making glucose, this G3P is the molecule that makes all of our organic macromolecules. When we identified our proteins from our amino acids, and we identified our sugars and our nucleotides, which make up our nucleic acids, or our lipids here with our long tails, all of this, all of the functions, all of the structures that are associated with all of our organic macromolecules, we're only able to make all of these because Rubisco can capture carbon dioxide and make it into the initial building block organic molecule that makes up everything else. If we did not have enzymes, especially Rubisco, life would not exist as we know it. And all of these enzymes, we have all these examples. I spoke a tremendous amount of a foreign language called science here, but the big idea is that you can remember a couple examples and we can tie it into building things up and breaking things down. When we put all of that together, we have metabolism. And that is it for enzymes at this point.